welcome to our quick dive into protein synthesis and metabolism. We're going to break down some pretty important stuff: how proteins are made, how they get broken down, and how amino acids are made and used in bacteria. Don't worry, we'll keep it chill and simple, like chatting with your science buddy. Let's kick things off with the first step in protein synthesis. Here's Simon to explain.
Alright, let's start with transcription. This is where DNA gets copied into RNA. In bacteria, an enzyme called RNA polymerase reads the DNA and makes a single strand of messenger RNA or mRNA. Instead of T or thymine, RNA uses U or uracil. RNA polymerase looks for a special DNA spot called the promoter, which is just before the gene. It finds this spot with the help from sigma factors. Here's the process. It starts with initiation, where RNA polymerase sticks to the promoter and opens up the DNA. Elongation then follows, where it makes the mRNA strand by adding RNA letters A, T, C, G one by one, moving along the DNA. Lastly, termination. When the transcription machinery reaches a stop signal in the DNA, RNA polymerase lets go, and a strand of mRNA is produced. Now, the mRNA is ready to be turned into protein.
Sweet! So now we've got our mRNA message, but how do cells read that to make proteins? That's where translation comes in. Kiara, you got this?
Yup, I got this. Alright, let's talk translation. Not like languages, but the kind your cells are doing right now to make proteins. Translation has three big acts. We have initiation, elongation, and termination. Think of it like building a protein sandwich, one slice at a time. Let's start with initiation. The small ribosomal subunit binds to the mRNA. In bacteria, it sticks near the Shine-Dalgarno sequence just before the start codon AUG. Then a special tRNA carrying the formyl methionine or FMET attaches to the start codon. The big ribosomal subunit joins to make the full complex. We now have a full ribosome ready to roll. Elongation time. Here's where the magic happens. The ribosome glides along the mRNA from 5' to 3'. tRNAs bring amino acids into the A site. The amino acid joins the growing chain at the P site. Then the ribosome moves everything along so the empty tRNA leaves from the E site. It's like a molecular conveyor belt. Add, bond, slide, and repeat until termination. Suddenly, stop codon. Could be UAA, UAG, or UGA. But here's the twist. No tRNA matches it. Instead, release factors bind, freeing the new protein, and the ribosome parts ways to be used again. And we're not done yet. Once released, the protein might fold or get chemically changed to work properly or be sent to the right places in the cell.
It's amazing to see how protein building works. But proteins don't last forever. They need to be broken down when damaged or old. Krisa, can you break down protein degradation for us?
You got it! Protein degradation is how bacteria control protein levels and recycle amino acids. Extracellular proteases are enzymes secreted by bacteria into their surrounding environment. Their primary function is to degrade proteins into smaller peptides and amino acids outside the bacterial cell. These smaller peptides and amino acids are transported into the bacterial cell through specialized transport systems, where they are used in biosynthesis for energy production. Protein degradation allows the bacteria to acquire nutrients.
Now, amino acids, or the building blocks of proteins, can be made in the cell too. Ethan, what can you tell us about the synthesis of amino acid?
One big process of amino acid synthesis is transamination. An amino group, NH2, is transferred from one amino acid to a keto acid to make a new amino acid and a new keto acid. This happens with the help of enzymes called transaminases or aminotransferases, which need a helper molecule called pyridoxal phosphate or PLP as a coenzyme. The process involves two parts. First, the amino acid reacts with the PLP attached to the enzyme, forming a temporary compound. Then this rearranges and breaks down to release the new keto acid and a molecule called pyridoxamine phosphate. This keeps cycling so the cell can make different amino acids as needed.
Great! Now, amino acids don't just get made. They also get broken down in different ways to keep the bacterial cell functioning well. Gyril, you're up with deamination.
Imagine this. An amino acid shows up inside a bacterial cell. The cell's like, cool, but what am I supposed to do with an amino group? Let us start with deamination, which basically means snipping off the amino group from an amino acid. This process is utilized when the bacterium needs to use an amino acid for energy. It is important to note that there is more than one way to deamine it. We have the oxidative and non-oxidative methods, but today we are zooming in on the oxidation aspect of deamination. In oxidative deamination, the amino group is removed. The resulting products include ammonia and a keto acid, which are important for metabolism. Certain bacteria like Enterobacteriaceae are really good at breaking it down. They use an enzyme called glutamate dehydrogenase, which removes the amino group and turns glutamic acid into a-ketoglutarate, which is important in the citric acid cycle. Also, aspartic acid, when deaminated, turns into CO2, ammonia, and acetic acid. These are useful as they are getting recycled, reused, and repurposed for energy and building blocks. That's the magic of deamination, cutting off of what is not needed and turning the rest into something useful.
Gotcha! Other processes are also involved in the breakdown of amino acids. Let's have a quick run-through of these reactions.
Decarboxylation is a process by which CO2 is removed from the COOH group of an amino acid. This is catalyzed by the enzyme decarboxylase. As a result, smaller reactive molecules like cadaverine, putrescine, tyramine, tryptamine, and histamine are produced, which help bacteria adapt, communicate, and survive in challenging environments.
Cool. So, we've removed the amino group with deamination and snipped off the CO2 with decarboxylation. You'd think bacteria would take a break, right?
Not at all, AG. We also have dehydrogenation, which is about extracting energy. In this process, hydrogen atoms are removed from amino acids, but they are not just tossing them away. They are capturing electrons and sending them to NAD+, or FAD, which then turn into NADH, or FADH2. These are energy carriers that power the electron transfer chain, helping bacteria make ATP. ATP, or adenosine triphosphate, is their energy currency. You will see this in action during glycolysis, the TCA cycle, and especially in aerobic bacteria like E. coli or Bacillus.
Now, person, we also have desulfurylation. What is it about?
Desulfurylation is exactly what you think it is. It refers to the removal of sulfur, usually in the form of hydrogen sulfide gas, from sulfur-containing amino acids such as cysteine and methionine. The key enzyme involved in this reaction is pyridoxal phosphate-dependent desulfurase. Most PLP-dependent desulfurases specifically act on cysteine, since it is the main sulfur donor in cells and has a readily accessible thiol group. Desulfurylation results in the production of hydrogen sulfide, ammonia, and pyruvate. Pyruvate, a keto acid, is essential in energy production or biosynthesis via glycolysis and the Krebs cycle. Amazing, right?
Indeed, it's amazing that amino acids get broken down in many ways. Last but not least, let's talk about why all this matters. What exactly is the importance of protein metabolism?
Bacteria don't always have a steady supply of food, so they need to use whatever they can find in order to survive. That means pulling the nutrients like the proteins, carbohydrates, and lipids straight from their surroundings. When it comes to proteins, the breakdown starts with the proteases, which chop those big proteins into peptides. Then those peptides get broken down even further into individual amino acids. Once the bacteria have these amino acids, they start processing them through amino acid catabolism. The first big move, deamination. Remember from earlier? It is the removal of the amino group, which is the NH2. This produces ammonia, which can either be excreted or recycled, and keto acids, which are super important for metabolism.
These keto acids do not go to waste. Bacteria funnel them into major energy pathways. Some enter glycolysis, where they are converted into intermediates like pyruvate. Others head into the Krebs cycle, which is the major hub for energy production. In the Krebs cycle, bacteria efficiently break down amino acids, releasing carbon dioxide and generating high-energy carriers like NADH and FADH2. These vital carriers are essential for the electron transport chain, where they create a proton gradient that powers ATP production through oxidative phosphorylation. Furthermore, bacteria leverage amino acids to synthesize crucial proteins necessary for enzymes, transport mechanisms, structural integrity, and regulatory processes. This comprehensive approach to protein metabolism is not just important, it is critical for energy production, growth, and survival.
Perfect! So, to recap, transcription copies DNA to mRNA, translation turns mRNA into protein, proteins get broken down when they're damaged or old, and amino acids can be made or broken down in several ways. All these processes are key to the life of a bacterium. Thanks for watching! We hope you enjoyed learning about these amazing processes. See you next time!